All right, the clock has struck 9. Good morning, and thank you for being present either in person or on Zoom. I am delighted today to introduce a guest speaker who is being co-hosted by both the Liver Center and the Nutrition and Obesity Center at UCSF, as well as the Division of Endocrinology. Most of you know her by name, Dr. Elizabeth Speliotes from the University of Michigan. We have to end quickly at 5 minutes before the hour, so I'm going to keep my introduction very brief. So Dr. Speliotes was actually born in Greece. She actually moved to Boston and spent her formative years in Boston, was a Yale undergraduate, and then moved over to Harvard, MIT, where she got an MD, a PhD, and an MPH. And then went through internal medicine and gastroenterology and joined the faculty at Mass General. She had an opportunity to move, and in about 2011, she moved to the University of Michigan to be an assistant professor of medicine and bioinformatics, where she has risen through the ranks to full professor and an endowed professor. So I think many of you know Dr. Speliotes. Her name is actually synonymous with the genetics of obesity and its metabolic complications. She has an enormous contribution to this field. And she is also head of the MSTP program at the University of Michigan. And I'm going to leave my comments there and allow her to have the floor to introduce us and really educate us on all the work that she's done in the genetics of metabolic diseases. Dr. Speliotes.
Great. I'd like to thank Dr. Mayer and everybody for being here today and for inviting me to tell you about some of our work. So these are my disclosures. So today what I'll do is give you an introduction to obesity, MASLD, and human genetics, and then tell you about our population genetic, functional genetic, and translational genetic studies in this space. So as many of you know, obesity affects many people. More than 66% are overweight, 33% are obese. It affects over 1 billion people worldwide. Accounts for 17% of our healthcare costs. And for today's talk, I'll be using BMI as a measure of overall obesity, waist to hip ratio as a measure of abdominal obesity. And right now we have an incomplete understanding of the pathophysiology of obesity, which gives us few effective treatments. Now, concomitant with obesity, you see an increase in the rise of related metabolic diseases such as diabetes, insulin resistance, dyslipidemia, hypertension, heart disease, as well as a metabolic dysfunction-associated steatotic liver disease, which is the new name for MASLD. But not all obese people actually develop these complications. And when they do, different people get different complications. But right now in medicine, we actually treat everybody the same. We say diet, exercise, take all these things. And really what that ends up doing is causing high personal and medical costs. And so I think, in my opinion, it's very critical to define the actual causes of obesity and its complications so that we can improve the diagnosis, care, and reduce costs the way we give iron for iron deficiency anemia and B12 for B12 anemia. And then similarly for MASLD, as people become obese, you can have steatosis that can lead to MASH. This can lead to cirrhosis and HCC. And, its global prevalence is about 24%. And its prevalence is actually higher in Asian individuals or Hispanic than Europeans and Africans. And again, we have few medical treatments and an incomplete understanding of its pathophysiology. And the other interesting thing is, just like obesity, as people become obese, many of them, but not all, will actually deposit fat in their liver. So if you take bariatric patients, 10% of them will actually not have fat deposited in their liver at all. And we think that about 20% of these may go on to get this inflammation, 20% will get the fibrosis, and 20% will get HCC. But we don't know who is going to progress to what. And actually, it looks linear here, but it's actually not that linear. You can actually progress to HCC without having to go through fibrosis. And similarly, in work that I did in the Framingham Heart Study many years ago when I was a fellow, we found that individuals that had more than 30% steatosis, 66% of them actually had this thing called the metabolic syndrome, which is having high triglycerides, low HDLs, visceral adiposity, insulin resistance, and hypertension. And some of these develop cardiovascular disease. And in work that I did with one of our fellows, Jessica, we showed that actually hepatic steatosis was associated with cardiovascular disease, even independent of metabolic syndrome. So even though I draw this and a nice simple arrows go here and there, we actually don't know who's going to deposit fat and go on to get liver disease versus metabolic disease. And that's really what we're interested in. And to try and do this, we have used human genetics. And and all of these things, so BMI, waist to hip ratio, MASLD, insulin resistance, are all common genetic conditions. And to measure that, we use a heritability, and these are anywhere from 26 to 60% heritable in the population. So that's you and me. And what that means is that unlike Mendelian diseases, where the one gene has a high penetrance and everybody gets the disease, here these common variants interact with the environment, and the right combination of predisposition plus the environment equals disease. But the reason why we stuck to human genetics is if we can identify genetic factors that associate with disease, they causally influence the disease. So your genome came before any phenotype you could have developed. So by definition, we can use that to understand susceptibility, and in particular, your susceptibility, define personal risk, and develop tailored therapeutics. And really, as Dr. Mayer said, that we're best known for our GWAS studies on body mass index, waist to hip ratio, liver function test, MASLD, and insulin resistance. And the way these work is we take genetic changes across the genome, and on the X is the chromosomes and the genetic changes. On the Y is the association with any phenotype. Here is BMI. And you can see these peaks coming out from the gray background. And these are areas of the genome that associate with a disease of interest. Here it's BMI. And just getting to this point actually has taken 2 decades of innovation with multiple people, not just me, getting the genotyping platforms unified, the phenotypes harmonized, QCing the data, and really getting large enough sample sizes to be able to find these peaks. But from these peaks, we now have over 9,000 of these peaks that associate with BMI, waist to hip ratio, more than 400 with LFTs and MASLD, and in our recent paper, more than 300 for insulin resistance. So again, it's a look into the disease that we've never had before. And then when we get these little peaks, so this is a zoom into one of these regions, again, on the X are these changes or the SNPs, and on the Y is the association with disease. You can see that it's actually not just one SNP, but a little flurry of SNPs. And that the reason for that is that your genome is not completely random. So these things developed in your genome, and so they are in something called linkage disequilibrium with a lot of other changes. And so we usually get these little peaks, and you can take the top SNP and you can drop anchor. Here, maybe for the people online, you can drop anchor, and then you can look around to see what genes are there. But a lot of times, when you drop anchor, you don't land in a gene, you land near a gene. And so there's still a little bit of like wiggle room about, well, what does this do and which gene does it affect? And so there's still more work to be done from these peaks to actually get to the gene, to the function, to figure out the biology. And so to help with that, we've developed methods, and here is one of them where what we did with Toon and Luda is you can take these genome-wide association studies and drop anchor on either side and look at the genes underneath them. And what they did is that they took gene expression data sets from tissues, and they basically created principal components in those. And then they mapped genes to those principal components, and then they mapped known pathways to those components. And that allowed us to sort of take a lot of these genes, the majority of the genes in your genome, we have no idea what they do. But if they kind of go up and down with other genes in your genome, you can say, oh, this is implicated in lipid biology or obesity biology. And so that gave us a lot more power to anchor these in the biology. So then we can take these genes under the GWAS signals and then say, well, what tissues are they expressed in? And maybe that's the causal tissue for the disease. What gene sets are they representing, and tell us what pathways we should be targeting? And do these genes sort of link up together, like is gene A and gene D part of the same pathway? And that way we can prioritize genes, potentially they're the causal genes in the region. So this was a huge advance, and it allowed us to take all those hits that we got and then map them to tissues. So for example, when we did this for overall obesity, which is BMI, we got the nervous system out. And then when we looked at pathways, it was overeating, energy metabolism, neuronal biology, but the majority of pathways were novel. And then we did have targets like MC4R, which we knew was a positive hit because we know monogenic mutations, thanks to Christian and other people here, actually affect obesity and cause childhood obesity, but now we had common variants that affect you and me near this gene that also affect obesity. And in fact, when you look at the common variants, those guys are actually taller than usual because they have hypogonadotropic hypogonadism. And so they knew that from the monogenic hits, and we could see that also in the common variants. So again, the phenotype matched between the rare genetics and the common genetics that we were looking at. In addition to that, we got other genes like GLP-1s, and of course, as you now know, these are now some of the big blockbuster drugs that work because we know the right pathway was hit, right? And so basically pharma is using the human genetics to validate targets that they go after. And so, our data actually was used and is being used right now to prioritize targets. But it also created new targets like GRP and the calcitonin receptor, that at the time nobody was going after, but now people are going after the calcitonin receptor because, again, people have done, we said, what calcitonin, how does that have to do with obesity, right? But actually there's neurons in your hypothalamus and the appetite control regions that actually express the calcitonin receptor. And when you knock these out specifically in mice, they do have an appetite effect. So even though, we label things using certain known pathways or known biologies, these things could be doing other things. And that's really the value of the discoveries that we're making and applying to the biology. The other thing is that when we initially hit this and hit the nervous system, people said, oh, yeah, it's behavior, right? So, these fat people just need to eat less. And we're showing that because it's in your brain. But really what we're showing is that we can map this to your brain. So if it was all volitional and in your control, we would never be able to map this to your brain. So actually it's biological, and we're finding out the biological susceptibility to these illnesses, which is what we need to alter to be able to change the ultimate outcome. So again, this also has sort of implications for some of the things that we kind of blame our patients and victims for their disease. So when we did this for abdominal obesity or waist to hip ratio, actually we got a different biology. So again, it's showing you that your method is very specific to what you put into it. So here we got adipose tissue coming out, we got mesoderm development, cell growth and survival, glucocorticoid receptor signaling, and targets like PPARG. And as you know, so some of the targets that are being used for obesity and targets like PPARG are now being applied to fatty liver disease as well. So a lot of these metabolic illnesses interact with each other. And then here, one of the messages that's come out from the abdominal obesity is that there's something called lipodystrophy that can cause a lot of disease. And so we usually talk about that in the rare genetic space, but really there's like common lipodystrophy as well, where people don't have the ability to expand, for example, subcutaneous adipose tissue, or the function of that is not proper. And then in those cases, you get more fat deposition in your kidneys and your liver, and that can cause sort of organ disease. So that's one of the biologies that's come out of sort of the fat distribution data that we have found. And then when we did this for MASLD, we see that liver, intestine, pancreas, and adrenal are enriched, lipoprotein metabolism, triglyceride metabolism, arachidonic metabolism, and targets like PNPLA3 that now people are going after. And again, different disease, different genetics coming out, and different pathways. And finally, in our most recent paper, where we looked at a measure of insulin resistance called the triglyceride to HDL ratio, we found that, again, that maps to adipose tissue, and the biology is more decreased adipose amount or function, more insulin signaling, cell growth and metabolism, and targets like GRB14 are coming out of this. And in fact, when we looked at this triglyceride to HDL ratio and insulin resistance, the overlap with the genetics of metabolic syndrome was greater than 92%. So in my opinion, I'm not sure that I can separate insulin resistance from metabolic syndrome, and maybe they are the same thing, at least genetically. Okay, so the other thing that we did is establish non-invasive tests for MASLD. So when I was a fellow, basically what I did is I approached the Framingham Heart Study and I said, I'd like to use your CT scans that you did for cardiovascular disease to measure fatty liver. And they said, nah, you can't do it. We only have a few slices. And so I developed a method where you could actually measure fatty liver disease in these slices, and fatty liver is darker than normal liver when you see this. And you can make this a quantitative trait. And based on that, I started a consortium on that CT-based measure. And then more recently, we have data from the UK Biobank. And so here they actually have MRIs with PDFF measured for fatty liver. But at the time that we were doing this, they had like 43,000 MRIs, but they only had 10,000 measures. And one of the things we've learned for genetics is that you need power. So basically what we did is we downloaded the MRIs and then used convolutional neural networks to teach it to read the liver and to measure the liver fat. And then so we were able to get very good measurements of the liver fat using convolutional neural networks in 43,000 individuals, and then could do a GWAS on that. And then we combine that with ICD-based MASLD from the UK Biobank, from the eMERGE Consortium, and from FinGen to really get more power to find hits that associate with MASLD. And you have to use some special statistics to combine a continuous versus and a dichotomous trait, which we did using a fixed effects meta-analysis. And then you have to refine the phenotype because there's a lot of other liver disease that sort of creeps through based on sort of misdiagnosis along the way. But when you do all that, we had 17 genome-wide significant independent variants that associate with MASLD. And when you look at those, we wanted to ask, well, for across ancestries, is it really the effects that are different across ancestries, or is it the allele frequencies that vary? And overall, I would say the effects actually are very similar across the ancestries, and it's more the allele frequencies that vary. And you can see that, in some populations, some of the effect alleles are just less frequent than in other populations. And so, and in fact, when you add up all the variation explained by genetics, we actually have more genetic predisposition in individuals of Hispanic ancestry than European ancestry and African ancestry. And in fact, it's really the genetics that accounts for more than 70% of that prevalence difference that you see in the population. So then we also look to see other sex-specific effects. And some of our variants, like PNPLA3, actually have a stronger effect in women than men. And some of our other variants, like TM6, have a stronger effect in men and women. And we don't know why, but we see this and we can document it. The other thing is we wanted to use something called Mendelian randomization. So once you get these signals, you can say, well, tell me if the genetic of obesity is causing MASLD or is MASLD causing obesity. So you can actually do that directionality. And so when we did this, we can show that MASLD is causal for fibrosis and cirrhosis of the liver and esophageal varices, which any hepatologist would say, duh, right? So, okay, so that worked. And then in terms of the other way around, it was really waist circumference and BMI that are causal for MASLD. And not surprisingly, meds that decrease your BMI work for MASLD, right? Like GLP-1 agonists. So again, you can sort of see that coming out of the genetics. And then probably one of the more exciting things that we found, which I found 10 years ago, but that's getting more and more refined, is shown on this slide, which is probably my most favorite slide in the whole slide deck, which is that, our studies look to see that these variants associated with steatosis. So all our variants here are aligned to the steatosis increasing allele. And then you can see that some of these variants predispose to cirrhosis, but some don't. Some of them predispose to higher serum LDL, myocardial infarction, and triglyceride, but some of them actually protect against that. Some of them associate with increased diabetes, but some actually protect against diabetes, and some associate with increasing obesity, but some protect against that. And you say, well, why is that interesting? Well, epidemiologically, I told you that fatty liver associates with obesity, diabetes, heart disease, cirrhosis. So this is what you expect epidemiologically. But here you don't get that. And in fact, here you get more what you see in real life, which is that some people develop the liver disease, some people get the metabolic disease. The problem is we've never been able to figure out who's who. But now we do have the ability to figure out who's who. Basically, using these genetic biomarkers, you can identify people that are more predisposed to liver disease than heart disease and maybe diabetes. You can identify people that are more predisposed to liver steatosis and more MI and diabetes, but not, and this is more of a lipodystrophy phenotype here, right? And not only can you identify these subtypes, but now you can link them back to targets that you can actually target to actually alter the course of that person's fate, right? And so this is why this information is very important to pharma, right? Because now you can see what targeting these genes would get you. And in fact, you can see not only the on-target effect on steatosis, but potentially side effects that these medications might have. And so again, this is why pharma is using a lot of this human genetic data to sort of guide their targets. And the other thing that you're going to have to take my word on is that the other thing that genetics has showed us is that if you can make weak antagonists to all of these effects here and add them up, basically what happens to these side effects is that they all go away. So combination therapy actually allows you to have a very strong on-target effect with all the side effects going away. And that's really where I think medicine is going. And in fact, some of the combination therapies for obesity are showing us that, and that they're getting better and better efficacy with fewer side effects when they do these combinations. And then in addition to that, when you look at these subgroupings, or partition polygenic scores, as we call them, we can see actually new biology coming out. So one of these subgroups basically affects genes that affect the release of triglycerides into lipoproteins and get it out of the liver. So problems with this retain triglycerides and cause fatty liver. Other genes sort of recycle back triglycerides to the liver and have high lipoprotein input as their cause. Other of these genes, like FTO, which is really this IRX3 and 5 gene, affect the fate of your adipocytes so that instead of being white, they can be more brown and sort of dissipate heat rather than store it as energy. Other genes play a role in either peripheral or liver insulin resistance. Others play a role in absorbing fats from your gut. And others play a role in changing glucose to fatty acids using de novo lipogenesis. And probably this last group, and really the hallmark of this diversion group, is that you have lower serum triglycerides, but higher cholesterol. And I don't know what this group does, but my best guess is that somehow it takes triglycerides, retains them in the liver, and causes phospholipid or other changes that may cause damage within the cell. But either way, this is all new biology and really giving us a resolution into the disease that we've never had before. And this is very exciting because, again, our goal is to try and get to what's happened in like anemia and other fields, where if we can get to the cause of your disease, like iron or B12, we can give you iron or B12 very specifically, rather than give you all these other therapies that are going to cause you side effects. So then what we also did is we took our 17 genome-wide hits and we looked to see how well they predicted your risk of getting MASLD, cirrhosis, or ACC in a totally different independent data set. This is our Michigan Genomics Initiative, where we have over 80,000 people now genotyped at the University of Michigan and linked them back to their EHR data. And what you can see is that these polygenic scores identify 10%, 5%, and 1% of the population that has an odds ratio of anywhere from 2 to 3 to 4 for developing these diseases. And really the only thing I know here is just your genotype. So again, this gives us new biomarkers where I can figure out who is going to be predisposed to getting these diseases just from your genotype, and it gives us new markers for better risk stratification of individuals going forward. So the other thing that genetics showed us is, as I told you, it showed us that, the biology is probably more in lipoprotein, triglyceride, and arachidonic metabolism. What I didn't show you that we and others have shown is that a lot of the hits that we're getting for non-alcoholic overlap with alcoholic liver disease, and also that a Mendelian randomization of steatosis really does lead to more advanced liver disease. And all of these things taken together have sort of moved us away from this idea that fibrosis is really the only important thing in MASLD. Actually, it's the metabolic disease that's causing the MASLD that's really important, and also moved us away from this non-alcoholic and alcoholic distinction into this new entity called MASLD, which I think fits better with some of the genetics. And so basically from a population genetics, this is my summary. So basically using GWAS, we've shown that BMI can affect appetite regulation, energy metabolism, neuronal biology, and identified targets. Waist to hip ratio affects mesoderm development, cell growth, survival, and glucocorticoid receptor signaling and targets. MASLD affects lipoprotein, triglyceride, and arachidonic metabolism and identified targets. And insulin resistance or metabolic syndrome affects lipid homeostasis, brain processes, adipogenesis, insulin growth, and gives you targets. And then our work has helped establish CT as a non-invasive test for MASLD. And now, as many of you know, we're using imaging more than biopsy to define the disease. That MASLD is heritability across ancestry, that the allele frequency more correlates with prevalence than the effect sizes. Some of the variants have sex-specific effects, And MR and steatosis does say that that's causal for advanced liver disease, and waist to hip ratio and obesity measures are causal for MASLD. And MASLD genetic subtypes predict different sets of disease outcomes, and that we have different MASLD genetic subtypes, polygenic scores, and that the etiology of MASLD is metabolic disease. All right, so in addition to doing these GWAS studies, you can get a sense of which genes are implicated, but in the end, it's still an implication. And in my mind, you still have to go and do the functional studies to really test that the things that you think are happening really are happening. And so you can do this gene by gene, but really what we're doing is sort of doing that genome-wide. And in particular, what we're doing is genome-wide CRISPR screens. And the way these works is you can take CRISPR and the guides and use lentiviruses to infect cells, and then you can basically identify cells that have increased steatosis or decreased steatosis by cell sorting. And then you can use next generation sequencing to get back the guides to show which guides increase steatosis or which decrease them. And then we go from that and we make sort of more targeted libraries that then we follow up in both HUA7 and HepG2s to find ones that reproducibly associate with a phenotype. And then we do detailed phenotyping using high content microscopy. And so doing that, basically what you see here is a log fold change for increasing steatosis, decreasing steatosis, and you can see that there's definitely certain genes in the genome that increase and decrease that. That includes genes like in our GWAS, like PNPLA2, that help release triglycerides from those lipid droplets. Therefore, a loss of function of that would cause the lipids to be retained and to increase fatty liver, as well as genes like DGAT2, which affect de novo lipogenesis. So if you're not making new lipids, again, you're going to have less steatosis within the cells. So we've also taken some deep dives into some of the biology of some of these genes, and some of these were in non-coding regions. So let me tell you one of my favorite stories about one of those. So we found that non-coding variation around this long non-coding RNA associated with actually those CT changes that I told you. The only thing is we found that the risk allele actually associates with increased liver attenuation. And increased liver attenuation is not fatty liver disease. So it's got to be either iron, glycogen, or something else, but we didn't know what that was. And really, we suspected glycogen. And the reason we suspected glycogen is from the human genetic data, the variant actually increased the expression of a nearby gene, PPP1R3B, that does play a role in glycogen disease, or at least supposed to play a role in glycogen. And so we saw that in two different data sets. So the human genetic data was telling us maybe this is glycogen, but again, we had to prove it. So we used CRISPR-Cas9 to actually make a very directed deletion right at this LOC. And again, in the cell lines, you could see that elevating PPP1R3B and also having high glycogen in these cells. And in fact, we were able to define a distinct biochemical signature. And in addition to that, the other thing we learned from these studies is that this deletion, this variant actually affects every single component of metabolic syndrome. So it increases your triglyceride, decreases your HDL, increases visceral adiposity, increases insulin resistance, increases hypertension. So really the poster child for metabolic syndrome, but it actually protects against heart disease, right? So again, we're able to separate the biology, right? So in my mind, metabolic syndrome may actually be very tightly linked to sort of excess glycogen deposition in tissues, and here in particular, the liver. And so that, and the other thing that this is showing is that for a long time, people have associated HDL with heart attack, but as you know, targeting HDL does not affect heart attack. So again, this is actually one of the top hits for HDL levels being low. And so again, this very nicely shows you that HDL is not really linked genetically with heart attack. And so this would tell you to not go after that as a target for heart disease because it's not linked. And so it also shows us that the non-coding variation in human really is causal for having disease. And in this case, this is sort of a common glycogen storage disease. We've also looked at this in mice. So one of the genes that we hit was LYP, LAL1, and basically variants around this decreased abdominal obesity in females and a decreased hepatic steatosis, MASH, and fibrosis. So we made a mouse knockout of this, and what we found is that the female mice actually had lower weight than their wild type counterparts. And the males had a little bit of that phenotype, but not as strong as the females. This was not due to a difference in food intake or energy expenditure. And by NMR, you could see that they had decreased fat. And when you dissected out the fat depots, you could see that these animals had reduced white adipose tissue, but not brown adipose tissue, and also decreased deposition of fat in their liver. The other very interesting thing about this is that this was diet dependent. So you saw this phenotype when you put them on a high fat, high sucrose diet, but you did not see this phenotype when you put them on a chow diet. So basically, this is a gene by sex by environment model that may mimic sort of the human biology and gives us novel insights into the disease. And in particular, using these mouse models, you can potentially better dissect out this new gene environment interaction. So for many of you that don't know, our rates of obesity were actually pretty flat until the mid-1980s, and then something happened after that. But there's a lot of debate about what exactly causes that. But basically, using these models, you can now sort of not only look at the biology of LYP, but also you can look at the diet, about what parts of the diet are really promoting this phenotype. And I have my own biases about what I think that'll end up being. But you can use these models now that it sort of seems to represent an evolutionarily conserved pathway to figure some of these things out. Okay, so basically from the functional studies, I hope I've shown you that we can use CRISPR to identify genes that affect MASLD, that non-coding variation affects PPP1R3B, which affects glycogen and really causes metabolic syndrome, but not heart attack, that non-coding variants can cause human disease, that LYP can affect adiposity, MASLD in sex-specific ways in humans and in mice. All right. So now for our last part, so translational part. So as I told you, we mostly work on common human disease, and common disease is genetic predisposition plus environment. So we are also interested in figuring out what in your environment is really causal for getting these diseases. And here, what we did in our consortium is we took our genetic variants and we looked to see what they could interact with to cause MASLD. And in particular, what we did is we looked at BMI, waist to hip ratio, serum lipids, serum glucose, serum insulin, basically anything that we had across our consortium. And what we found is that actually at least PNPLA3 interacts most with insulin resistance to cause your MASLD, and more than glucose, triglyceride, or BMI. And specifically what you see here is the different genotypes of PNPLA3. The GG is the risk phenotype. And you can see that individuals that have the risk phenotype and high insulin, their rates of getting MASLD are over 90%. But if you don't have the risk genotype and you don't have insulin resistance, your rates are about 20%. The other thing that you see here is that the slope of this line here changes from the low insulin to the high insulin. And this is what's called a multiplicative effect in gene environment interactions. And what this means is that 1 + 1 doesn't equal 2, 1 + 1 = 8. And you're really looking for these things, because if you have the genetic susceptibility and the wrong environmental factor, wow, your disease risk increases. And this is very common in genetics. And you want to find these things because if you can reverse this, actually the people most at risk have more benefit than the people down here, right? So that's exactly what precision medicine is all about, right? And so that's what this would implicate. The other thing is, as I told you, we found that insulin resistance more than glucose sort of drives this effect, and people were just confused about that because for a long time we've known that diabetics have a higher rate of fatty liver disease, and people say, well, glucose defines diabetes, so what's your problem? Like, you got the wrong answer, right? Glucose is your problem. But actually, glucose isn't your problem, right? So there's something about the insulin resistant state that really promotes MASLD. And in my opinion, that probably has to do with a change in energy utilization, where you use a lot more fatty acids, probably. I can't prove that to you, but basically those fatty acids do circulate more. They do come to your liver to cause the fatty liver disease. But it's not glucose, right? So glucose is not as causal as the insulin resistance or something related to the insulin resistance that causes MASLD. And again, that requires a little bit of a shift in some of the paradigms and the things that we teach people, because I think people sort of blame glucose a little too much for some of the pathophysiology that we're seeing in these conditions. The other thing that was important about this study is that we actually did it in pre-diabetics. So we excluded the diabetics, we did it in pre-diabetics. And so all of the data basically shows that pre-diabetics who are not getting treated for anything, and also people that have this GG phenotype, are extremely high risk of fatty liver disease. And that's about 8% of the European population right now. And it's actually a larger fraction of the Hispanics and the Asians, and a slightly lower amount in African Americans, because the allele frequency of that actually varies quite a bit. So again, this is a lot of people that really are super high risk that we're just not doing anything about right now. And really what this data shows is we probably should be doing something about that, because they're really high risk. And so towards that, we look to initiate some clinical trials. So with Laura, we basically recruited individuals that had a PNPLA3, either GG or G, from our biobank, and then put them on a keto diet that decreases their insulin resistance, and supplemented that with online teachings, monitoring, text reminders, and coaching. And basically at 4 months, these individuals lowered their steatosis by 53%, their BMI by 12%, and their insulin by 41%. And many, about half actually really like the program and we're not going to stop. So again, what this shows us is that a low-carb diet really does reduce MASLD, even in the most genetically predisposed. And so we're looking to do a larger sort of randomized control trial based on this, to show that this is really an alternative sort of treatment for MASLD that will work. So in addition to that, as I told you, we did that study, the gene-environment interaction, showing that these diabetics that had GG really are at super high risk of MASLD. And so here we extended that. And what we did here is we looked to see, how well genetics sort of predicts insulin cirrhosis over time. And in particular, for those of you that are not in the liver field, what we do right now to risk stratify people for MASLD is we can do this non-invasive score called the FIB4. And if you have a high FIB4 score, you come to, hepatology, and we monitor you much more closely for liver disease, do treatments that are more aggressive because you have more advanced liver disease. But if you're intermediate or if you're low risk, we basically say, go away, come back when you're more diseased. But really here, what we did is that, we looked back, and here's the Michigan Genomics Initiative, what we also did it in the UK Biobank. We found individuals that had increasing liver function tests. And then we followed them over time. And basically the people that have intermediate FIB4 diabetes, but no PNPLA3, had this red risk. The people that are high FIB4 that are coming to hepatology right now are here at blue. But look at these guys. These are the people that have intermediate FIB4. They have diabetes, but they have PNPLA3. Their risk is way higher than even the high FIB4 guys, right? So really, these guys need to be coming to hepatology for us to be doing stuff for them, right? So really, this suggests that genetics really should be used as part of our better risk stratification guidelines to be able to define sort of a group of individuals that we now consider low risk and bring them into the high risk category so we can get them care before they develop the advanced liver disease. Furthermore, we also looked to see whether the genetics could actually affect response to medication. And here what we did in our Michigan Genomics Initiative is we looked to see how people that were on semaglutide and also had the PNPLA3 affected their change in ALT. So, and what you can see at 6 months, 1 year, and 2 years is actually the people that had the GG or the CG actually responded better to semaglutide and dropped their ALT more. So again, gene-environment interaction, right? So just like I told you before, if you can find out what's wrong and you can reverse sort of the environmental factor, the people most at risk are actually going to benefit more. And that's what this study shows. So really, what I've shown you is that insulin resistance multiplicatively interacts with genetic risk to cause MASLD. Ketogenic diets can reverse steatosis in genetically predisposed. Genetics and diabetes reclassifies people from intermediate risk to high risk, and that genetics can affect how people can respond to medications. And we probably should be using that for precision medicine. So really, I hope I've shown you how we can take sort of human phenotyping, use the genetics to find the cause of disease, find the environmental factors that interact with that, model them in cell and animal models, and then bring that back to patients for further stratification and potentially new precision treatments for them. And so what other stuff are we doing? So we're going more to whole genome sequencing now to look more for rare genetic variants that predispose to these traits. We're doing some single cell and spatial transcriptomics, CRISPR screening, as I mentioned to you. We're looking more at obesity subtypes, gene environment interactions, ancestry specific effects, and then also doing a little bit of work in the characterizing variants of unclear significance. And so with that, I'd like to thank the many participants in our studies, our many partners in the consortia and biobanks that we work with. I'd like to thank our departments, NIH, and other funding sources. And I'd like to thank all the people in my lab who I learn from every day. And thank you for your time as well. Thanks.